What's up, all my Diamond Hand soldiers? We have another presentation today for Applied Genetic Tech Corporation. Make sure to like, subscribe, and turn on your notifications because you don't want to miss out. Ticker symbol AGTC. Gene therapy is the process of modifying a patient's cells by adding a functional copy of the mutated gene, which is causing the patient's disease. The new functional copy allows a patient's own body to produce proteins to treat or even prevent genetic conditions. A single treatment can provide long-lasting benefits, potentially leading to a better quality of life. Now let's take a look at their balance sheet. Their total current assets are 82,964,000, and their total current debt is 12,963,000. Now, if we divide these numbers, we get 6.4, which is great for this company. Investors usually look for this number to be over 1 to see if it has a strong financial stability. Now, let's see what institutions are invested. We have Stonepipe Capital, currently its largest institutional holder, holding almost 4 million shares. That's almost 10% of the company. We also have BlackRock holding just under 2 million shares at just about 4% of the company. Let's take a look at what analysts have to say about this gem. At this time of this recording, AGC is at $5.30 per share. We have a high price target of $35 per share and a low price target of $14 per share, and an average price target of $22 per share. That's over 600% upside for the high price target and 300% upside for the average price target. All right, guys, I want you to watch this short clip I provided, and remember, diamond hands.
AGTC is using gene therapy technology to develop long-lasting therapies for people with severe diseases. Our initial focus is in ophthalmology. Genetic defects in over 200 known genes have been associated with poor visual function. Genes are segments of DNA that direct the cell to produce proteins that perform a vast array of functions in the body. Many diseases have a genetic basis, meaning that mutated genes provide incorrect instructions that cause the cell to produce an abnormal protein, or disable production of a protein completely, either of which can cause disease. In gene therapy, a functional copy of the gene is delivered into a patient's own cells. Normal protein that is produced from a functional gene has the potential to correct the underlying cause of the disease and provide restorative therapeutic benefit. Gene therapy uses engineered viruses to deliver genes into cells. These are called viral vectors. Vectors made from adeno-associated virus, or AAV, are especially well suited for treating retinal diseases. AAV is a small, simple virus that elicits only a weak immune response and has never been shown to cause disease in humans. In gene therapy, the two native AAV genes are removed and replaced with DNA that contains a functional gene with instructions to produce a normal protein. The DNA also contains a promoter, which drives production of the functional protein. The promoter and gene are flanked by signals called inverted terminal repeats that allow the DNA to be packaged inside the protein shell of the AAV vector. AAV vectors are delivered to cells in the retina by routine ophthalmic delivery methods, such as intravitreal or subretinal injection. Once administered, AAV vectors enter the patient's cells and deliver a copy of the functional gene directly into the nucleus. Once inside the nucleus, the functional gene can support the production of the normal protein and correct the underlying cause of the disease. Genetic defects in the eye can cause vision loss or blindness, and gene therapy can provide a long-lasting therapeutic benefit. AGTC is using its technology to develop gene therapies for rare, inherited retinal diseases that currently have no treatments. One of these diseases is achromatopsia, an inherited retinal disease which is present from birth. Achromatopsia results in markedly reduced visual acuity, extreme light sensitivity causing day blindness, and complete loss of color discrimination. The retina is a layer of cells at the back of the eye. In a healthy eye without achromatopsia, special cells in the eye, called cone photoreceptors, convert light into an electrical signal, which is transmitted across the layers of the retina and then to the brain for visual processing. In an eye with achromatopsia, cone photoreceptors in the retina are unable to convert light into an electrical signal. The most common causes are mutations in one of two genes, called CNGB3 and CNGA3. In gene therapy, an AAV vector containing a normal CNGB3 or CNGA3 gene is designed to provide the instructions for normal CNGB3 or CNGA3 protein and restore cone photoreceptor function. By introducing a functional copy of a gene into a patient's cells, gene therapy is able to correct the underlying genetic defect that is causing disease. The functional gene, delivered by an AAV vector, is thought to remain stable for many years, which suggests that gene therapy has the potential for long-lasting clinical benefits after a one-time administration of the vector. AGTC has a significant intellectual property portfolio, expertise in the design of gene therapy products, and expertise in the formulation, manufacture, and physical delivery of gene therapy products. For more information, please visit www.agtc.com.